Good afternoon, everyone, and welcome to today's Seminars in Oncology. We are delighted to have with us today, actually live in Boston, Dr. Patricia Stieg from the National Cancer Institute and the NIH. Pat has been a leader in cancer biology for many years, particularly in the area of breast cancer biology, and we're interested to hear of her latest work. So by way of introduction, Pat received her undergraduate degree at the Towson State University in Maryland, followed by a PhD at the University of Maryland College Park. Subsequently, she went to the NIH, where her scientific career blossomed, and she not only is a leader in, scientifically, but as well as within the organization of the NIH and NCI. She currently is the deputy chief of the Women's Malignancies branch in the Center for Cancer Research of the NIH, among other titles and accolades. So as I said, Pat has done path-breaking work, and today she'll be talking to us about brain metastasis of breast cancer. So Pat, welcome, and I'll turn it over to you.
Okay, thank you. All right, you see my presentation, right?
Not yet.
Not yet.
Why not?
Share your screen.
I already did once. Heavens.
Need to practice Zoom for 2 more years.
Okay, how do I get to share screen again?
Do you have at the bottom of your screen?
At the bottom of your screen, yeah.
Let's go there. Oh yes, how about that? Okay, let's try this again. Tell me it's working.
Perfect.
Yep,
You can
it's
just go into
working.
presenter mode.
Okay. Find my laser pointer and I'm off and running.
Perfect.
Well, good afternoon, everyone. Thank you, David, for the lovely introduction. As he mentioned, I'm a couple of miles away at the convention center for the Society of for Neuro-Oncology meeting. So if you have questions, you're welcome to contact me and come on down to the hotel or something. Okay, this is going to be a new seminar, so you're all going to have to bear with me, but I hope you're interested in the data. And it's a different aspect of brain metastasis, concerns brain metastasis when diagnosed in young patients. So we typically say a patient's young when they're 40 or under. And those diagnosed young have far more advanced disease and are very aggressively treated. And despite this, their survival is poor. There are many multivariate analyses. I'm going to show you some of them. They have adjusted for everything you can think of. I've listed them on the slide, and a young age is still an independent predictor of poor outcome. And this has been, you know, you have things that stick in the back of your head as to why. And at some point, you decide on a testable hypothesis and see what you can find. And quality of life concerns may be very distinct in these young patients. They're young, they're either having families or want to have families, they have careers, a lot in front of them. Okay, this shows just 5 of maybe 15 multivariate analyses that are out there right now about breast cancer when diagnosed in young patients. The top one, I believe, is from Ann Partridge, who is part of the Harvard faculty. 17,000 in this cohort, all breast cancer patients. And mortality, if the patient was diagnosed under 40, the hazard ratio is 1.4. And she found significant correlations to the luminal A, luminal B, and triple negative subtypes. You come down to another one, 300,000. And again, if you look just chop at less than 30 to 59, so you're going to the younger end, breast cancer specific mortality was greater. A triple negative study, this I think is Carrie Anders, And if under 40, disease-free survival and overall survival was lower, this was only triple negative patients. So it says something about that subtype and nothing else. This is Carrie. Early breast cancer. And you can see that I can't add because I have 2 lines that say 3. If from cohort either under 45 or over 65 and disease-free survival was lower, you're getting the idea. Here is a young cohort, 18 to 39. And if they were at the bottom end of this, 35 and below, survival was lower, and it was lower in the non-Hispanic, white, and African American populations. And finally, an early breast cancer population that were all premenopausal, age less than 35, poor disease-free survival, overall survival compared to any older. Increased local regional relapse, metastasis, and they saw a nice correlation in the luminal B and a trend in the triple negatives. So what I think you can see is everyone agrees that a young age is a hazard, and the younger, the more hazardous it is. And I don't think there is a consensus if there is one subtype or multiple subtypes that are the bad actors here. This is a study that Alex Zimmer in our branch published a couple of years ago now. We work with Walter Reed, National Naval Military Center, and they're literally across the street from the NIH, and they see a lot of patients. And because it's a military outfit, they have a lot of young women as recruits into the military. If they get breast cancer, they know it's aggressive, and they often transport them up to Walter Reed for their treatment. And as well as these patients then have one health system. They're all in the VA military system. So you could take that variable out. And they also have a higher percentage of African American and Latin Hispanic recruits. So we start to get some numbers on those populations. So this is basically what we found. The blue bars are under 40, and then 40 to 49 and greater than 50. If you look at the stage at diagnosis, there was less stage 1s in the young population, more 2s and 3s. If you look at tumor grade, it's much higher, a lot of grade 3 disease. And if you look at the hormone receptor status, it's lower. And this is all very consistent with the literature. We were able to find out in this system what kind of treatment these younger patients receive. Breast conserving surgery is down, mastectomy is up, and bilateral mastectomy is up. Did they get chemotherapy by stage of disease? Look at this in stage 1 disease. These young patients were given chemotherapy, and they remained at high levels of chemotherapy. And if you go on to systemic therapy, this is interesting. If you look at the ER positive subtype, they all got hormonal therapy, depending, irrespective of age. But did they get hormonal therapy and chemotherapy? And here you can see the increase in the young patients. Overall survival, this is a cohort of 10,000 patients. What recurrence was higher, and the overall survival of the youngest and the oldest groups was poor. When we broke out the African American and Hispanic subgroups, there was no significant difference. And that's interesting now that, they've all had the same care. So breast cancer patients that were in the DoD database, when you diagnosed at a young age, underwent more surgery. They were given chemotherapy for less advanced disease. They were given combination therapy more often. And despite this, their survival was still lower than the mid group. So when treated in a uniform health system, the African American and Hispanic subgroups were not significantly different than the Caucasian. Okay, so that presents a bit of the issue. And now we get to starting to ask why. So the easiest thing to ask is there a smoking gun mutation in the primary tumors from young patients? And we looked at two databases, you see TCGA and Metabrick, and used the 40 as a cutoff. And all this is showing you is disease-specific survival. And to show you that 40 is a good cutoff. You see the difference in survival at 40. And here is the bad news. These are mutation frequencies in TCGA and Metabrick. There's very little overlap between these two. You do have a decrease in PIK3CA mutations in both, significant in one. Other than that, there really aren't many trends that can hold in either, and there's very few mutations that affect a good proportion of these tumors. Enter a wonderful postdoc, Alex Wu. He's left the lab, and I was joking with David before this. For those of you who haven't seen it, there is a video online that is a takeoff on Lady Gaga's Bad Romance or Bad something relationship, and it's called Bad Project. It's a science video. You need to go look at it because Alex would pull this up after he enthusiastically wanted to do this project, and it just mired. He would pull this up and play it from time to time. So there are other published comparisons of primary tumors from young and older patients. And there's nothing striking in methylation. There's nothing striking, as we said, in the subtypes. And the question we asked, and Alex was so initially thrilled with, is if it's not the tumor, is it the host? And so, metastasis is an interaction of tumor cells and host, and seed and soil and all that stuff. And so the hypothesis was that host factors determine, at least in part, the aggressiveness of breast cancer. So with that, we made a few decisions. We decided to use metastasis as an indicator of aggressiveness. And so what we compared was metastasis in old and younger mice side by side. And we used triple negative and luminal B models because of the data that you've already seen. And long term, what I would love to be able to say is there something that should be done differently for young patients in terms of therapies to make them more effective and work in a young body? So this is data that was done in collaboration with Lala G. Wakefield, and who is a wonderful metastasis investigator looking at the TGF-beta pathway. We pulled out here 4 different metastasis model systems. I believe they're all triple negative. The first one is MDA231, and it's a tail vein experimental metastasis in the tail vein. And how many lung mets do you have? What you see is that the differences in age for each experiment of what we're calling young and what we're calling old. And I'll explain that very quickly. We had 2 ways of acquiring old mice. That's one of the hardest parts of this project. One was to purchase retired breeders from the supply houses. And that way you'd get a jump on aging these mice. And we would age them, but then the question is, how do you do the controls? And what we did is we got the young mice in, put them through a round of breeding and involution so that we held parity as a constant. They'd all had litters. And so there you will see young as old as 6 months because it's the time to do the breeding and the involution. There are other mice that we just got in age. They just ran around in their cages very happily for forever. And then you could just get in a cohort of young mice and acclimate them to the facility, and you'll see those at 2, 3, 4 months. Okay, So that's the difference. But and old in a nude mouse, you are lucky to get past 12 months. They just get sick. Where in some of the syngeneic models, you'll see old at 20 months, which again, deserves an award for getting there. All right, so tail vein assay, MDA 231, and you can see no real difference in lung metastases. There were then 3 spontaneous models of metastasis where we injected the cells in the fat pad, grew a primary breast tumor, either resected it or didn't, and there's the data. So let me move this. There we go. EO771 went into old and young mouse. This first graph is the primary tumor weight. There's no real difference. And the number of lung metastases. This is a non-resection model, as is the MVT1. There is a small difference in primary tumor size, no difference in metastasis. And then in 60T1, it's a resection model. So the tumors were resected on day 14, nothing. We were not happy, but this is where the Brad Project videos started showing up. We also looked at brain metastasis. Simply, I have a lot of experience with it. We used 2 models that are well known in the lab and well published. One is the 231BR, it's triple negative, human, so it goes into nude mice. The other is the 4T1, and these have been through 5 rounds of selection, BR5. So we've injected them and harvested the brain metastases, grew them out, re-injected. And you can see that they make what we call metastatic colonies. These are on their way to becoming, they will eventually coalesce into one huge brain met that the neurosurgeons will tell you, oh yeah. We also derived EO771 BR5, triple negative, immunocompetent. You can see here the number of metastatic clusters that form, parent to BR2 to BR5, and you see that nice progression. I would be lying if I told you these were brain tropic. They are very, very highly metastatic, so you get more metastases in the brain. You may get less systemically, but believe me, they're still there. And from Johanna Joyce, we obtained the 99LN series that she was deriving for brain metastasis, and thank her. This is luminal B, and she advised us that it might need a few more rounds of selection. So we put it through 4 rounds. And what you see are nice metastases that grow out in this. So there's 4 model systems to look at brain metastasis. And here would be the data. So in every case, you see the ages of young and old. And in every case, these are cells injected at left cardiac ventricle that will spray the cells throughout the body, and you will get brain metastasis development. And to our surprise, there is a 3- to 4-fold increase in brain metastasis for model systems in young mice compared to older mice. Interestingly, to get to the specificity question, the 99LNBR actually makes one other kind of metastasis pretty well, and that's to the liver. Same mice, no difference in liver metastasis. So there's something going on in the brain. And this is where the Bad Project video started again, because you're asking, now what? I wanted to stop and quickly tell you that brain metastases preferentially develop in young humans. So here is a study of age at diagnosis of a primary breast cancer, how many brain mets, how many primary tumors, and you can go all the way to the right to the incidence. And you can see that it's highest in the 20 to 39 age group, and then just progressively goes down. So this is not just a mouse thing, this is real. Here's 2 other studies of 219 patients that died with metastatic disease under 70 at metastatic presentation, 22% got brain mets. It was related to young age, with 43% of women under 40 developing brain mets. And that's about where we're sitting. Of 2,248 patients, 7% developed brain mets over 54 months, age 35 or younger. HER2 triple negative were the significant factors. If they were under 35, it was independent of a biological subtype. That question just can't seem to get solved. Okay, so here are things we tried. They seem like obvious examples to explain why these young mice were getting more brain mets. It doesn't seem to be a blood-brain barrier permeability difference. We looked at blood-brain barrier permeability with Quentin Smith at Texas Tech. To paclitaxel or a marker didn't see any difference. We asked if, in a roundabout way, we asked if extravasation into the brain was different. What we did is we bypassed that and we put the tumor cells directly into the brain with an intracranial inoculation. And as you can see in the picture, the young ones still made larger tumors. There's a good reason to think about hormone levels, and I'll show you a slide on that later. But in mice, they don't undergo a true menopause, and the levels of testosterone, corticosteroids, etc., were all about the same. We looked at the neuroinflammatory response. So around a developing brain metastases, activated microglia and astrocytes will come in and surround that lesion. And we looked at the number of, here you have data on GFAP. These are the activated astrocytes. There is no difference. We looked at tumor cell proliferation, no difference. Tumor cell apoptosis, no difference. On comes the bad project video. What we think it is that more cells are just starting the colonization. Once they start, they're just all proliferating at the same rate and nothing's dying. Okay, and that brought us to the immune system, which was a subject I can't tell you I'm familiar enough with. And we teamed up with Dorian McGavern at NIDDK. He is a CNS immunologist that works in infectious diseases and was happy to take our metastatic mouse brains and do the cell sorting for us. And what you see here is the, how do I let's get rid of this. There we go. What you see here is the data from young and old mice. And these are BALB/c mice with 14-1BR, triple negative metastases. If you're looking at overall immune cells, there's a significant difference. Nothing much in CD4, nothing much in CD8 T cells. There's a difference in neutrophils. It didn't rise to significance. Monocytes, significant. Macrophages, significant. Microglia, significant. Higher levels in the younger. Now, that could be due to metastasis, or it could just be due to mouse age. So we did the exact same experiment. It was actually side by side on naive mice. So these are mice without any tumor. And here again, with age, is this difference. Of these, we don't see a difference in monocytes. We don't see a difference in macrophages. And the number of microglia, these are the innate immune cells in the brain, do decrease with age. This is well known in the literature. So you've got multiple changes in the immune environment, much of it in innate immunity, and some of it only in the metastatic setting. Despite the fact that it was only really the myeloid cells that showed significant differences, we worried that there could be minor differences in others of those cell populations that weren't statistically significant, but were biologically important. So we did antibody elimination studies. This is in the 14-1 model, and we used anti-CD4 to knock out that population, anti-CD8 to knock out that one, anti-GR1, knock out the neutrophils, etc. pre-treated the mice. And what you can see on the first graph is, did we knock it out? And it worked beautifully. And then we injected the 4T1BR cell line, no difference, no difference, no difference. So again, the bad project video comes on. What we did see was we used Plexicon 3397, which is a colony stimulating factor 1 receptor inhibitor, CSF1R inhibitor, and it will take care of most of those myeloid cells. We used it initially in chow, and it took about a month to deplete the myeloid cells. This would be the microglia as well as macrophages. And when we injected these cells, bang, we had a nice reduction in brain metastases. So what are we talking about? This is sort of a pictorial. Here is the normal brain. So you have a capillary, and this is the blood-brain barrier. Along the feet of it are astrocytes. Out in the parenchyma, you have the microglia. These are the innate immune cells. You have neurons, and the microglia spend their time touching all these neurons and these astrocytes and everything else to sort of monitor, is everything okay here? I haven't shown any infiltrating macrophages. It's a tough field. Okay, here is a sort of depiction of a metastatic brain. And I'm showing you two different pictorials. Over here on the left is the uninvolved brain. And what you have is basically what's over here. Okay, you have all the component cell processes. On the right, you have the brain metastatic microenvironment. So here are the brain metastatic cells. They grow on the abluminal surface of what's now the blood tumor barrier. They are surrounded by this neuroinflammatory response that I mentioned briefly. These are activated microglia, activated astrocytes, and in essence, they try and wall off a lesion. You find these not only in brain mets, but traumatic brain injury, any brain injury that you're talking about. You will also find some infiltrating, did I put something up there of them? No. Perivascular macrophages. They are poorly characterized. Our analysis of them is that they are primarily in the leptomeninges, the lining of the brain. But we did find a few that are sitting along the blood-brain barrier capillary. So they really cannot be ruled out. They're just very tough. Now, we asked how the Plexicon reagent worked. First, by looking at IBA1 myeloid cells. So this would be microglia, macrophages, etc. And we looked in both compartments. We counted in the uninvolved brain, and we counted in the metastatic microenvironment. And you can see a decrease with Plexicon there and a decrease with Plexicon in this metastatic microenvironment. If we wanted to center in on just the microglia, we use P2Y12, and here you can see much nicer decreases. So it shows you that the agent worked. It worked the same in young and old. When we compare young and old, I'm sorry, that was just young. When we compare young and old, we went to oral gavage of this so that the mice's potential peculiar eating habits wouldn't get in the way. And again, started the oral gavage and injected 4T1 cells. And you can see a nice reduction. It's a strong trend. It's just tough to get these animals. In brain metastases in the young, the old are low to begin with and don't get significantly lower. And so you have a preferential effect in the young. Again, when we counted to say, how did this work? I can tell you offhand that it worked the same in young and old. Here you have young in the uninvolved and old in the uninvolved region. And these are all myeloid cells. Here's the young and the old around the met. And here's the microglia aspect of that data. So the young mice have greater numbers of myeloid cells than old mice. That is apparent from our initial self-sorting and in the literature. The Plex reagent showed a strong trend in preventing brain mets, only really in young mice. It removed comparable percentages of myeloid cells and microglia and from old and young brains. Microglia seem to be removed a little better. And again, we're asking that general question, well, now what has happened? So there are a few online descriptions of microglia, characterizations from old and young mice. And we availed ourselves of those papers and started to ask, did we see any of these in our metastatic model? The one that rang true was a surprise. It's semaphorin 3A. Now we think of semaphorin 3A as a neuronal protein because it is involved in neuronal axonal growth and patterning in development. But here is a picture of metastases from old and young mice. These, I believe, are 4T1. IBA1 is in red. So let's go here and you can see that there's plenty of myeloid cells, microglia, as well as macrophages in these lesions. SEMA3A is sort of gray. And here you can see the young is these myeloid cells are replete with SEMA3A. These myeloid cells, no. And so there is a picture of the difference that we saw with age in the metastatic microenvironment. And here you can see some quantification of it. So as I said, SEMA3A is known in axon growth cone regulation. And but there is a literature that it has effects on microglia as well as some cancer cells. So as usual, things do more than you thought. In vitro, if you add SEMA3A to a Boyden chamber assay, it will increase tumor cell motility. It will also increase in vitro tumor cell proliferation. So we think there is good preliminary evidence that overproduction of SEMA3A by these young microglia in the metastatic microenvironment might be one of those signals that is stirring these tumor cells to proliferate and move. In the uninvolved region, we saw a different pattern. SEMA was barely made in the uninvolved brain. What we do see, and it's been well known, is that old brains, even if there's no insult, will have activated astrocytes. And so here we're looking at GFAP, and just look in the control and you can see that there's more GFAP positive astrocytes in a perfectly, uninvolved brain than young, okay? When you take these mice and treat them with a CSF1R inhibitor, so here you have in uninvolved more GFAP positive astrocytes than in the young. With Plexicon, this goes up in the young, and it goes up to levels that are not far from what the old normal is, and they go up even a bit more in the old, and you can see that over here. Nothing happens in the metastatic microenvironment. There you've got the neuroinflammatory cells. It's full of activated microglia and astrocytes. You're not going to get more GFAP there. And so now we come to my grand cartoon of this project. So in the young normal, I showed you this picture. In an older normal brain, what you've got are less microglia. You've got some activated astrocytes, and that a little bit of SEMA3A being produced. In the young metastatic brain, again, we looked at this cartoon. You have less microglia in the uninvolved brain, okay? And you have this huge neuroinflammatory response. And in this mix, a lot of SEMA3A is being made, okay, which can act on these tumor cells. In the older, you have less of the SEMA3A. You have more activated astrocytes. And so you do not have, these are potentially inhibitory to metastatic colonization, and you have a reduced amount of something that is potentially stimulatory. But here's the take home, that CSF1R inhibition, it does preferentially prevent brain mets in young mice, but it gives a young brain characteristics of an old brain. They make fewer metastases, they have more activated astrocytes, and they have less SEMA3A. And it's just it seems to phenotypically age the brain. I'd like to know down the line, does it have neurocognitive effects? So the investigations continue. We still want to know what cells are responsible. Are these microglia or infiltrating macrophages? And we want to know more about CSF1R inhibition. The Plexicon reagent, one of them made it to the clinic. It's not the most potent or the most specific, and we're working with other ones now. And we're clearly asking, does it have an effect without a pre-treatment? Because I don't think you can do that to a patient. Does it have a preventive effect, or will it treat an established brain metastases? Is it preferentially active in the young patients, alone or rational combinations? And as I said, I really want to look at neurocognitive effects, because we seem to be aging these brains, or phenocopying aging. At night, as I'm laying awake, I want to know why we didn't see anything different in liver or lung, and are there other host factors operative with age and breast cancer? And if I had to choose one, it would be response to treatment. We could do the whole thing over, but I don't think I have the nerve to start this over again at this point. Now, what we saw in the myeloid cells with the CSF1R inhibition is not going to be the only host factor that's operative. I've had the pleasure of working with Diana Citelli, who's at the University of Colorado, and she has published a couple of very good papers on the effects of estrogens. And to do so, she used triple negative models. So they are unresponsive to estrogen. They're estrogen receptor negative. The question is the host estrogen responsive? And to do what we saw, no differences in estrogen levels with age. To more precisely mimic the age of a human, what she did was ovariectomize all her mice. She either then treated them with a vehicle, she gave them estrogen, or she gave them letrozole, an additional suppression, okay? And this is the 4T1BR model. You're looking at micromets and metastatic colonies. Come out over here, it's just very easy with the vehicle and very little, ovariectomized. You got very few metastatic clusters. If you added estrogen, you got incredibly increased metastasis of a triple negative cell line, and letrozole further kept things low. Here's the survival, and it's in favor of the ovariectomy. Here's what you're looking here at, a different model system, EO771, and you can see, again, with the estrogen, it's high, and with letrozole, it goes down. Here in the panel on the right is what she has figured out. She did not center on the microglia. She centered her work on the astrocytes, which are ER positive, and they respond to estrogen. And she's come up with a lot of very interesting pathways that concern BDNF and EGFR to reciprocally stimulate those triple negative breast cancer cells. So the estrogen may be acting indirectly on these triple negative cells via activating these astrocytes. Fascinating work. And we continue. I'm just going to take you briefly through work that we are doing at the proteomic level. We're doing this with Tom Conrads at the Inova Medical Center, and he's part of the Walter Reed National Military Center. What we did, and this was started a long time ago. So we started with the human 231BR triple negatives into nude mice. If I were doing it today, it would clearly not be. It would be syngeneic in young and old mice. At day 30, we perfused the mice to get everything out of the bloodstream and collected the brains. And for proteomics, what we did was a massive amount of laser capture microdissection. So what we did is we took part of the brain, and here is an H&E. The first thing we laser captured out were the tumor cells. We then took a so many nanometer area around that colony and called it the microenvironment. And then we went to the same area in the opposite hemisphere that had no cancer in it and called that uninvolved brain. So we had 3 different types of microdissected material: tumor cells, tumor microenvironment, and uninvolved brain. So there are about 1 million ways to analyze this data, and I can tell you we've been down through a few of them. What we've come up with, the paper is being prepared now, is what we first asked is what proteins were specific. We were most interested in the microenvironment, the metastatic microenvironment to go with. We were first interested in what proteins were concentrated in the metastatic microenvironment compared to uninvolved. So, you've got the wealth of proteins in uninvolved. Let's see what is up or down regulated in the metastatic microenvironment. With that, we compared that list between old and young. And so we ended up with 3 different piles of proteins. Microenvironmental proteins that were upregulated only in young, microenvironment proteins that were upregulated only in old, and those that were upregulated irrespective of age that came through. Here is the ingenuity pathway analysis, and it tells us that we've got a lot of work to do. We have not touched this part of it, potentially very important. The brain metastatic microenvironments in the aged mice have altered metabolism, and it just comes through in multiple pathways. This is the part that we've started in on, and that is the age microenvironment is less permissive for cell movement and migration. They're things I'm familiar with. So this shows you some of the proteins that came out in this analysis, and I'm just going to show you preliminary data on tenascin C, TNC. So tenascin C is very well known in the metastasis literature, although there isn't a literature on its effect with age. It's an extracellular matrix component. It has EGF-like repeats, fibronectin domains, a fibrinogen domain. It exists as trimers and hexamers. It is anti-adhesive, pro-migration, and pro-proliferation. And by anti-adhesives, it interacts with syndecan-4 to prevent fibronectin from binding. And it can also be an integrin ligand. It's found in the normal brain. It's elevated in primary tumors and secondary microenvironments. It promotes metastasis. We wanted to know what it's doing with age and brain metastasis. And we're still after this. We started a lot of these validation efforts by doing immunofluorescence, thinking that that would give us the most specific information. That certainly might be true. What we ended up getting our best data from was just taking 41 metastatic brains from young and old mice and crushing them up. I would have told you this would not have worked. The metastases were too small. They occupied such a small part of the brain. but it was much easier than dealing with the vagaries of immunofluorescence and different antibodies and their specificity. What you can see is here's the tenascin C band, and these are ground up metastatic brains. And you can see if 4 of the 5 have a very good signal, and the 5th, with imagination, it's there. In the older brains, it's 3 out of 4, it's not there, and 1 with imagination. So you see the trend that the proteomic analysis saw. If you just take naive brains, it's not as clear. You see the tenascin C in the young, and you see it in maybe half of the old. So there is some validation. We wanted to start looking in vitro, and of course, there's quite a literature out on tenascin C, and we chose migration studies. And these are with 231 and 4T1 cells. And so you make a scratch at 0 hours, and we looked at the tumor cells filling that scratch over 24 and 48 hours. Now, you can put tenascin C in, and you can see that it will fill in the scratch. But if you think about how these tumor cells are actually growing, they're growing on the outside of the blood tumor barrier. And that has the parenchymal basement membrane or astrocytic basement membrane, which is replete with collagen and laminin. So I wanted to know how it stacked up against good old type 4 collagen. And it looks pretty much the same. However, you put the 2 together, you start seeing something. Okay, and that is the answer here. So I can't tell you that tenascin C is a more potent mutagen than typical basement membrane collagen, but that it adds to it. And that's what we tend to have found. We've done tenascin C knockdowns in the 4T1s, and that has many of the same, and the 231s, and it has many of the same effects on migration. So we're just at the beginning of understanding the effect of patient age on metastasis to the brain and to other organs. This work is difficult. It is not for the faint of heart. The ability to get healthy aged mice is just tough. Difficulty in nailing down the host factors once you see something, and then being able to see differences that you think might relate to the human. We still wonder why only brain metastasis was affected by mass age. And again, I think there's going to be other host factors. I also wonder if when we analyze primary tumors for mutations, we really didn't look at metastatic lesions, and there could be hits there. I'm hoping that the proteome will provide a more complete picture of what's going on in that metastatic microenvironment. And I'm hoping that some of these leads will lead toward an age-specific therapy. All along in this study, I have been guided by members of the Young Survivors Coalition. This is a breast cancer coalition of patients diagnosed when they are young, and to listen to what they're going through and what they worry about. And so there are many, many thank yous. Brunel Grill has been a constant component in the lab, but she has left. And for those of you who write metastasis grants, she's joined NCI program and is leading their metastasis grants. Imran Khan has been in the lab, and Alex Wu was the brilliant postdoc who has left. Alex Zimmer is an oncologist who's been an integral part of this. Quentin Smith at Texas Tech worked on the permeability. Dorian McGavern was the amazing immunologist that helped us. Biology Wakefield with metastasis assays, Tom Conrads with proteomics, and other people at Walter Reed, including Craig Schreiber. And with that, I will thank you and take questions.
Well, thank you very much, Pat. That was phenomenal. And you're right, it is such a challenging area, but you addressed it so rigorously. I did like the allusion to that Lady Gaga Bad Romance.
You all have to go find that video and realize it got played almost weekly through this project.
But again, I was actually just listening to a talk where the main point was that it's really only worth doing things that are hard. And this is something hard, but you're really getting at a core question. And let me ask a broader question, perhaps, while people use the Q&A functionality. We're now learning in other tumors that interaction between neuronal cells and the tumor cells, you get this bidirectional effects. We've seen that now in pancreatic cancer, even in hematologic cancers in the bone marrow. Do you think this is a larger area that has been unappreciated?
I do. In brain mets, Josh Neiman at UCLA has been studying this. And it's like the work we've been doing, where they're just on the cusp of understanding what's going on. And I would love to see it expanded. In one of our studies years ago, we looked for neuronal cell death, which is not always easy to quantify. And I think we used fluorojade staining or something like that. And I would like to see it expand to not only, the typical vicious cycle of, they attach to neurons and neurons make things that the tumor cells use, but are these neurocognitive deficits that are so endemic to brain metastases, are these interactions killing neurons, and how big of a cone of death are you getting, et cetera?
Well, I think there are a lot of interesting points. So let me actually go to one of the questions that is coming in, which actually relates to this in a different way. Do primary brain tumors or their metastases elsewhere in the body, though rare, have any similarities to the results you show? So you're talking about breast cancer metastasizing to the brain. How about primary brain tumors? Any thoughts about that?
I spent the first part of the pandemic on my living room couch writing a review for the blood tumor barrier. And I got bold enough to include primary brain tumors, which I know something about, but not much. And a lot of the literature overlaps, because correct me if I'm wrong, in the primary brain tumors, the radiation oncologist and the surgeon are going to get the bulk of that tumor. And what you're dealing with is the invasion of those cells out, be it on the capillaries, the neurons, etc. And there is a literature for the CSF1R inhibitor in GBM. And in fact, it's the Novartis compounds in trial in GBM. So I think there are similarities, particularly if we set up models for GBM that really focused on the invasion part and not just plop 100,000 cells down and see if that spot grows.
Yeah, no, that's a great point. And actually, when you're talking about the blood tumor barrier, let me just ask your thoughts about this. That's always so controversial because people say, oh, well, you have to develop therapies that will get into the tumors. And other people say, well, the clinical hallmark of metastases to the brain is contrast enhancement, which generally represents contrast leaking across. So any thoughts about those?
Oh, do I have thoughts, David? Thank you. I will send you a check for answering that question, for asking it. They are diagnosed by gadolinium imaging. It's a very, very small imaging agent and a very, very sensitive imaging analysis, okay? When we look at permeability of brain mets, and we've looked at it to markers and 5 different drugs. This is with Quentin Smith and Paul Lattman. What we see is that 85% of the brain mets are more permeable than normal brain. They would have all lit up with gadolinium, okay? 15% we're not finding, but there they are. However, some of these brain mets get this much of drug or marker in them, others get this much, others get a whole lot. What we found is, so what we did is we looked for the amount of marker of drug, we did it first with paclitaxel, that has to get in to cause an apoptotic tumor response, any apoptosis, okay? Only the 10% of lesions that had the highest amount of drug in showed an apoptotic response. So yes, there's more permeability, but only the highest amount of permeability had an effect. And when you compare those amounts to the amount in systemic lesions, you're off by a log. I rest my case.
Well, that's good. That was a very quantitative answer. I mean, and that I think is an important consideration.
Now, people are making brain permeable drugs. I mean, it's not impossible, and they're getting approved.
Absolutely. Let me we only have a few minutes left. Let me kind of go back to some of these questions. There were some reports suggesting that PARP inhibitors may inhibit microglia activity. Any suggestions from early PARP inhibitor trials that they may prevent or treat brain metastases in young women?
None that I'm aware of is the short answer. We tried them in young mice because that's what we routinely use, and didn't see a lot. And I'm unsure of their brain permeability. I think they fall into that category of a regular drug that I just described.
All right, well, then maybe I'll follow up with a question from Rakesh Jain that actually I was going to ask about.
Hi, Rakesh.
About the ER positive aspect within the brain. So do ER positive breast cancer patients receiving letrozole or similar agents have fewer brain metastases?
Yes. Yes.
So there's clinical data that would suggest that.
I mean, brain metastases predominate in the triple negative and HER2 subtypes, not in that very voluminous ER sub. Now, give them long enough, you're going to get brain mets. So by then you've got resistance, etc. But yes, I think we've done this experiment.
And so
somebody put this in trial.
Okay. Well, let me go to another question. I was going to follow up on that, but so there's a question about more brain metastases as opposed to lung or liver metastases in the 4T1 BR cells in the young group. And the question relates to whether you can generate lung liver specific cells by injecting and collecting the 4T1 metastases in the lung or liver over several cycles. And will one see a phenotype in the lung liver metastases instead of brain metastases?
It's a wonderful question. Go for it. We all had good luck in the brain and we all had good luck in the bone of generating these tropic lines. I served on a study section where every grant was how to make liver specific models. And I heard a lot of ideas, but I still don't see a lot of papers. I don't have a good answer for this.
And let me maybe circle back for a moment about the I give you credit, you kept talking about the neurocognitive effects, which people, again, that's another hard question to look at that people have not looked at rigorously. Any other thoughts about that in terms of the brain metastases themselves or the various therapies that we're using that may affect these cell types to lead to neurocognitive decrement?
I can tell you what we're doing because it is when you ask the patients what they care about most, they care about their cognition. You hear horrible stories from the oncologist of the decline, particularly after whole brain radiation therapy or a bunch of SRS. What we have done, and it ties into one of your other questions, is we are borrowing from the primary brain field. So we are working with Terry Armstrong, who works on neurocognition. And she was at MD Anderson for years and years. And we've managed to pull her into NCI. And she has developed 2 patient-reported outcomes. One for the ability to get through the daily tasks of the day, and the other of cognition, okay? You do it on an iPad, you do it while you're in the waiting room, each time you're there. We have been running a phase 1 trial. It's going to get reported out at San Antonio to look at secondary prevention of brain mets. And the success that we had just giving, these were developed as indicators of brain tumors, of the effects that brain tumors could have. So it seemed like a perfect idea for us to go to brain mets. I mean, of the times we put, a patient sat down in the waiting room, I think we had 97 or 98% compliance in filling out these evaluations. And we've got numbers. And I think, you can say, oh, it's biased because it's self-reporting, but the ability to do it and get the data, I think is a wonderful idea. And I mean, what I can say is these numbers change with progression. They change, that graph goes, it might be progression systemically or in the brain, and we've had to start parsing that out when we get larger number of patients, because this was a phase 1 trial. But I think that's the way to go, or at least to combine it with some of those other tests. I don't know.
Well, this is very impressive, going from really very basic and mechanistic questions all the way to human beings. So I really want to applaud all of your contributions over the years, including this very recent work, which is so exciting.
Thank you so much.
We are at 5 o'clock, so I do want to conclude. I want to thank Pat again for coming and sharing her great work with us, and I look forward to opportunities to collaborate going forward.
Thank you so much. Everyone have a great day.
Okay, bye-bye.
Bye-bye.